want to spend enough time, kind of stop at 1:15 here, to introduce you to the organization. That's what we started off with the nervous system. All right, what were the four basic tissues of the body? Blood is connective. Bone is connective. Epithelial, nerve, which we just finished, and now we're doing the fourth one, which is muscle. Okay, we're going to continue to look at nerve not as a tissue. We've done the cranial nerves. We'll go back and visit some of those, like the facial nerve and the hypoglossal nerve, with our skeletal muscles. And as we move through the rest of the body and do muscles, we're going to associate that particular terminal spinal nerve with it, so that you'll learn what nerves innervate the muscles in the anterior arm versus the radial nerve. There's all the muscles of the posterior arm and posterior forearm. And I find it easier to learn the nerves with the muscles that they regulate than to keep them completely separate. So I blend this together. Physiologically, when we come back from Thanksgiving break, we learn the physiology of how a muscle cell contracts. We're going to take that process of an action potential being sent down the axon terminal, and we'll follow that down the lower motor neuron, where it causes the synaptic boutons of the lower motor neuron to release acetylcholine. And acetylcholine is going to bind to receptors on the skeletal muscle cell, which are nicotinic receptors. And are nicotinic receptors ionotropic or metabotropic? Ionotropic. So they are excitatory, which is what we would expect on skeletal muscle. All right, so that part we know already. And now we're going to see on the acetylcholine binds to an ionotropic receptor on a postganglionic neuron. It forms graded potentials. The voltage is a ligand-gated channel. And so if enough sodium ions move into that postganglionic neuron, it triggers the threshold of the axon of the next neuron in sequence. Same thing with muscle. When acetylcholine binds to its nicotinic receptor, which is going to open sodium ions, if enough sodium channels flood into the muscle cell, it will create an action potential that will spread along the skeletal muscle cell deep into the skeletal muscle cell and open calcium channels, just like it does in a neuron when it moves down the axon terminal. All right? So that part is very familiar to you. And the new part will be what do those calcium ions cause the skeletal muscle to do, and how does it shorten. So that functional part we'll pick up when we come back from break. Today I just want to look at the overall kind of organization and some of the terms. They can be confusing because they all start with similar words. And our prefix for muscle utilizes two, myo and sarco. And both of these mean muscle. All right? Now if we look at a skeletal muscle, such as one of these muscles on our upper extremity, say the deltoid muscle, goes over your shoulder. It is surrounded by a sheath of dense regular connective tissue that encloses that muscle and separates it from other surrounding muscles. And those fibers come together to form a tendon. And we talked about tendons as a location of dense regular connective tissue. So here we have the bone. And remember, the bone is covered with periosteum, which is also dense regular connective tissue. So we have periosteum. And then blending through that and inserting to form Sharpie's fibers, we have the tendon, which is also dense regular connective tissue, blending into the dense regular connective tissue of the bone. All right? So these are going to come from the sheath that surrounds the named muscle. And so this band of connective tissue, when it's actually over the muscle itself, is known as the epimysium. Right? So this surface, if I'm touching a muscle itself, itself, it's covered by dense regular connective tissue, which is known as epimysium. Now, a lot of times in artwork, this is drawn as a series of concentric rings, but in actuality, those fibers are coming straight out at you, okay, because they're going parallel with the muscle bundle itself. Now, within the muscle, we have groupings of 10 to 100 individual muscle cells. And so those individual muscle cells are the groups of individual muscle cells are identified as a fascicle. So each circle that I'm drawing is going to be a single muscle cell. Our word for a single muscle cell is a myofiber. So there's a cluster, and again, these are cut in cross-section as well, because muscle cells are the same length as the muscle. So if I have a muscle going from my lateral epicondyle to my wrist, that's the length of the muscle cell. All right, so this is just cutting them in cross-section. That's why we're seeing them in a circle. So I'll draw another fascicle over here. So a cluster of cells is a fascicle. And the connective tissue holding them together is dense irregular.
Oh, that's irregular.
Dense irregular. This is known as perimysium. Epimysium is dense regular. And perimysium is dense irregular. So here would be the epimysium. Each of these is a fascicle, all right, and then around them is the perimysium. And then around individual muscle cells is loose irregular. Remember where we found loose irregular always underneath an epithelium? Because it provided a good medium for diffusion, small capillaries. Same thing around individual nerve cells and individual muscle cells. We have loose connective tissue. It supports the capillaries, nourishing those small cells. So inside the fascicle, in between each of the individual muscle cells, is the most loosely organized material, which is endomysium. So this model that I have up front is from a single muscle cell. This is the plasma membrane of one cell. It's kind of cut open here in cross section. Here's a motor neuron coming down to innervate it. So this is representing the loose irregular connective tissue around that single muscle cell, which is the endomysium. Okay. We only have one cell here, so we don't have enough to form a fascicle, and we obviously wouldn't have the perimysium or epimysium either. Okay, now I'm going to erase this in just a few moments, and I want to take one of these single muscle cells, one single black circle, and show it to you as a myofiber. Okay, so here is showing a muscle, and this would be the outer covering, the epimysium. Each of these clusters is a fascicle. And so around each of those is the perimysium. And then in between each of these is endomysium along with the small capillary supported by that tissue. So if you have a microscope at home, feel free to section up your turkey and take a look at it. Okay, so these structures are confusing in this list because they're shown almost the same size and everything looks like it's filled with little circles. So up here at the top would be what I drew here, one named muscle like the gastrocnemius or the deltoid. Here would be a fascicle, a group of muscle cells. So here is one of those fascicles, one of these structures. And then here, each of these would be a single muscle cell. Now, it looks confusing because each single muscle cell has little red dots in it as well. Okay? And that's because inside each muscle cell, here's one cell, a myofiber, and the little red dots are bundles of actin and myosin. Remember the microfilaments we talked about? So inside the cell, surrounded by the sarcolemma, the plasma membrane of the cell, we have bundles of actin and myosin. And those bundles cause these little circles inside the cell. So I want to start with this level now and erase the diagrams that I have on the board. And I'm just going to draw one cell.
So a perimysium is made up of a fascicle? Would that be?
The perimysium surrounds and bundles the fascicle.
Surrounds.
It's a wrapping around it.
Okay.
Perimysium is dense irregular. So we go from dense regular to dense irregular to loose irregular as we get around an individual muscle cell.
Wait, peristome is what?
Perimysium. Epimysium is on the surface of the muscle.
No, peristome.
Perimysium.
No, peristome.
Oh, periosteum. That's the
Periosteum.
dense regular
Yeah,
connective
that's right.
sleeve
Okay,
of the bone. Sorry.
sorry.
So if this top from there.
I say the way Rachel likes it.
Is the epimysium?
I'm not sure what this is supposed to be out
I'm
here.
sorry.
But yeah, that would be, this is probably, I don't know what that is. But
And
this
then-
would be the epimysium.
And then that's the.
And then this would be the perimysium, and this would be endomysium here.
Well, that's the.
Okay? All right, so let's look at one skeletal muscle cell, and then I'm going to draw it a little bit smaller. All right? So this is one muscle cell or a myofiber. And our word for the plasma membrane of a myofiber is sarcolemma. Remember how we had axolemma? So lemma means membrane, and the plasma membrane of a muscle cell or myofiber is a sarcolemma. So this is the plasma membrane surrounding the cell, and that special plasma, it's not special only because it's part of the muscle cell, but that plasma membrane of a muscle cell would be the sarcolemma. Okay? So the endomysium is outside the muscle cell. It's connective tissue. It's not part of the muscle cell. The plasma membrane that defines the muscle cell itself is the sarcolemma. All right, then inside the muscle cell, the myofiber, we have bundles of actin and myosin. Those are known as microfilaments, if you recall from our cell structure. So actin and myosin. Remember that was about 5 nanometer and 10 nanometer filaments. When we talked about the cell, we talked about them as microfilaments because of their diameter compared to microtubules and intermediate filaments. Well, these microfilaments, when they are inside a muscle cell, are known as myofilaments. They're still microfilaments as far as being a cytoskeletal structure, but we just call them myofilaments when they're inside a muscle cell. All right. So think about different sizes of spaghetti. Big fat spaghetti. My son's favorite, my youngest son's favorite is angel hair, okay, or the really thin stuff. And so remember, actin is the thinnest. is 5 nanometers in diameter, and myosin is 10 to 12, so that's going to be bigger. All right, so for every myosin filament, we have 6 actin filaments. So here would be another myosin. And there would be the actin. And here would be a myosin. 7. Here would be a myosin. So they can share them. Now remember, myosin has the one that was formed of golf, is that right? Golf club-like heads. And if we put more than one myosin protein together, we have this pattern. So on Tuesday next week, we'll talk about how they grab the actin and pull the actin towards the midline. So each of these myosin filaments is going to be able to grab onto those actin and pull them towards the midline of this band of structures. So we have actin organized like this. It fits into a disc. We'll talk about this structure on Tuesday. And then we have another actin, a myosin arrangement. And then this is fitting into a disc like that. So this bundle of actin and myosin, again, think about the spaghetti, all right, fat and skinny spaghetti, is found in clusters again inside the muscle cell. There is no, these are not enclosed within a membrane like a cell. I'm not trying to be precise here.
That's actin and myosin?
Actin and myosin.
The myofilaments.
So if we were to look at them, we would see this kind of a structure. Woodpeckers are alive.
Sorry.
All right, so this entire arrangement, what I've shown here, a bundle of actin and myosin, is known as a myofibril. I know, we're going from myofibers and myofibrils and myofilaments. So this would be a myofibril. This would be a myofibril. This would be a myofibril. Okay? I hesitate to draw a circle around that because there's no membrane actually around it completely enclosing it. It is associated with membrane sacs, but it's not like a lysosome or a mitochondria where it's completely surrounded by a membrane. So back to this model. Here would be a whole bunch of actin and myosin. Here's another one, and another one, and another one, and another one. Each of these is a myofibril. All right? So if we look at this diagram here, it's very similar to this model. Here's the plasma membrane, the sarcolemma, and each of these is a myofibril. Here's the myosin, here's the actin. The pink is the actin, the blue is the myosin. Now, yes, there is a membrane wrapped around them, but they're not completely enclosed in it, and that's what I'm going to get to next. All right? So this structure of the myosin and the actin, we'll talk about it in more detail on Tuesday, but from here to here, neither the actin nor the myosin will get shorter. What happens is the myosin pulls on the actin and pulls the edges closer to each other. So look at the distance between my elbows. From my elbow to my fingertips will be actin. And we'll assume that there's a band of myosin in between. Okay? So now the myosin, that invisible myosin, is grabbing onto the actin and pulling it inward. So look at the distance between my elbows. And now when the myosin grabs onto the actin and pulls it together, the two edges move together, what happens to the distance between my elbows? It gets less. It gets shorter. And that's how a muscle shortens. Neither the actin nor the myosin become shorter. But less space is taken up as the edges of the actin are pulled together. Okay?
I don't understand. I understand how the actin doesn't get shorter, because it's not really changing, but something.
The myosin is not getting shorter either. I need to find another ruler here. I